All right, let's get started with chapter 18. So, welcome to the last official lecture, Biochem. You survived, made it this far, so well done. And so we're going to do our last chapter today, which is chapter 18. So we'll start with the electron transport chain, and then we'll do some stuff on oxidative phosphorylation. So, let us begin with just what is the overall purpose of the electron transport chain? Yeah. Yeah, exactly. So we are generating a proton gradient from our reduced electron carriers. And what are we going to do with that proton gradient after the ATC helps make it? Yeah, we're going to use it to make ATP. So where do our reduced electron carriers come from? Yeah. Yeah, exactly. So all of those different catabolic pathways that we learned about, all of those help to generate all of these reduced electron carriers. So those are things like glycolysis, the pyruvate dehydrogenase complex, the citric acid cycle. Those are the ones we learned about here. There are also other ones that we did not cover in this class. Any questions? Yeah. Okay. Okay. I just wanted to confirm that we don't have to know the specific what's happening in 5 steps. We just have to know generally what becomes what and why. Yeah, exactly. So you don't, if it helps you to think about the mechanism, to understand what it does, that's great. You don't need to know the mechanism for the exam. Yeah, you need to know what you start with, what you end up with, where it comes from, where it's going, like where it fits in. But yeah. Yep. Yep. Any other general questions? All right. So let's go over the electron transport chain as a whole. So for the ETC as a whole, every one NADH that goes in, how many protons are moved in total? Yeah, 10, yeah. And where are they going from and where are they going to? Yeah. Yes, yeah, they're going from the mitochondrial matrix to the intermembrane space, which you can also call the IMS, because intermembrane space is long.
Really quick, can you explain where the 10 comes from? The 10 nitrogens?
Yeah, so let's actually look at the picture that illustrates it. So each of the complexes generates a particular number of protons. So if NADH enters at complex 1, and we'll go over this more in the later too, but so NADH starts with complex 1. complex 1 moves out 4 protons for every NADH, complex 3 does another 4, and then complex 4 does 2. So for SADH2, how many protons are moved? Yeah, it's 6, exactly. And we will talk more about that in the coming slides also. So yeah, so that's, now we have our proton gradient. And what is it being used to synthesize? Yeah, ATP. And what is doing the generating of ATP? Yeah, the enzyme. Yeah, so ATP synthase. But yeah, the process is oxidative phosphorylation. The enzyme is ATP synthase. All right. So now that we just quickly reviewed this, but how many protons are moved into the matrix by each complex? And so before we begin, I invite you to tell me what is wrong with the question that I'm asking you. Yeah, that is true, but there's something wrong with the question itself. I heard it in the front. Yeah, exactly. It's not moved into the matrix, it's moved out of the matrix into the intermembrane space. Okay, so now that we know which direction we're going, how many protons are being moved by complex 1? 4. Yep. And how about complex 2? Yeah. Complex 2 is not also 4. Yeah, it's okay. Yeah, it is 0. So complex 2 is the one that doesn't actually pump out any protons. But complex 2 is special for another reason that we will talk about on the next slide. So how about complex 3? Complex 3 is 4. Yeah, you're like one step ahead. It's 4. And then how about complex 4? That one's 2. Yeah. And just to be clear, that's how many protons for every reduced electron carrier that enters. So like for every NADH that enters, or for every FADH2 that enters, that's how many protons get moved. So, and depending which complexes each electron carrier travels through, tells you how many total protons each electron carrier will ultimately push or help to push through. So, which complexes are for which electron carrier? Yeah. Mm-hmm. Yeah, exactly right. So NADH and FADH both pass through complex 3 and 4. At the end, NADH enters through complex 1 and bypasses complex 2, goes straight to 3. FADH2 enters through complex 2, never passes through complex 1. And so that's where our different numbers of protons arise from. Because we talked about how complex 1 gives you 4 protons. Complex 3 gives you 4. Complex 4 gives you 2. Complex 2 gives you 0. Not my best 0. Okay. So, yeah, so why is NADH producing more ATP than FADH2? Which we kind of just answered, but tell me again. Yeah. Yeah, exactly. They pass through slightly different complexes. So NADH goes through complex 1. So ultimately it's moving more protons. And the more protons you have, the more ATP you can make. And we'll talk about why that is when we talk about oxidative phosphorylation. So that brings us to our direct connection to the citric acid cycle. So of our 8 citric acid cycle enzymes, which one participates directly in the electron transport complex? Yeah, F, that's right, succinate dehydrogenase. So this is, what's special about this enzyme? What electron carrier does it reduce? FADH2, yeah. And we just talked about how FADH2 enters the electron transport chain through complex 2. And in fact, this enzyme, succinate dehydrogenase, is part of complex 2. So the FADH2 goes straight into the electron transport chain that way. So during the citric acid cycle, FAD is reduced to FADH2. During the electron transport chain, FADH2 is oxidized back to FAD. Any questions about that? Okay. So let's talk about electron carriers. So I'll give everyone a few minutes to work on this on their own. So take a few minutes and think about these different electron acceptors or carriers, and think about how many electrons each of them can accept. And we'll come back and discuss it together in a few minutes. All right, let's start with NADH. So how many electrons can NADH accept? Yeah, 2. Yeah, so so we start with NAD+. It can accept 2 electrons. And how many protons does it then need to become NADH? Yeah, just 1. Yeah. All right, how about FADH2? Yeah. Yeah, so FADH2 can accept 1 or 2. For all the pathways we discussed in this class, how many does it accept? 2. Yeah. So FADH2 starts out as FAD when it's oxidized. It can accept 2 electrons. And how many protons to get to FADH2? Yeah, 2 protons. All right, so how about coenzyme Q? Yeah, 2 electrons. Yeah, so coenzyme Q plus 2 electrons. And how many protons? Yeah, also 2. Protons gives you reduced coenzyme Q. And coenzyme Q, similar to FADH2, can also accept just 1 electron. But for most of what we are talking about here, it's 2. All right, how about cytochrome C? Yeah, 1 electron. How many protons does cytochrome C accept? Yeah, that's right. It doesn't accept the protons because the protons get pumped into the intermembrane space. All right, and that brings us to oxygen. So for every O2 molecule, which is 2 oxygen atoms, how many electrons? Yeah, 4. Yeah, that's right. So O2 plus 4 electrons, and how many protons? Well, what is it going to eventually turn into? Water. Yeah, so 2 oxygens. We need 2 water molecules. How many protons do we need? Yeah, 4. Yeah. So, yeah, the oxygen one's a little bit weird because it's kind of a lot, but realistically, for most of these, we're talking about moving 2 electrons at a time. But because you never see just 1 oxygen atom floating around by itself, you need to double up everything when it comes to an oxygen molecule.
Can you just explain how NAD accepts 2 electrons but only 1 proton?
Yeah, so usually when it's written out as like a full equation, you'll usually actually see plus a proton because the extra proton just doesn't attach, like bond to the NAD molecule itself. It's just kind of there to balance out the equation. So like you could also write it like this as 2 protons on the left and an extra proton on the right. But in terms of the actual molecule, it's sort of helpful to think of it as just accepting the one proton, because the other proton just kind of floating around. Yep. And yeah, so let's think about this in terms of for every, I'm just going to change color. So for every one NADH that you reduce, how many coenzyme Qs can you reduce? So if you take the electrons from an NADH and you stick them on a coenzyme Q, how many coenzyme Qs? One, yeah, because there's 2 electrons from the NADH. 2 electrons can reduce now the coenzyme Q. How about cytochrome C? Yeah, it's 2 cytochrome Cs, because each cytochrome C can only accept 1 electron. So NADH donates 2. You need 2 cytochrome Cs to accept them. Right, and how about an oxygen molecule? Yeah, exactly. So those 2 electrons from NADH will only reduce 1 oxygen atom, which is a half of an oxygen molecule, but those don't actually exist in real life, which is why everything has to get doubled when it comes to oxygen. All right, so any questions about this mathy part? All right, so let's now that we know what all these electrons are, because it's also going to be donating too. So yeah, same idea. Yep. So let's think about how all of these electrons being passed around now fit into the electron transport chain into each complex. So we'll go back to our complexes slide. So which electron carrier is getting involved in complex 1? Yeah. So what's happening to NADH? Yeah, it's getting oxidized to NAD+, and then there's no really good way to, trying to think of a good way to write it, but what's happening to, what other electron carrier is taking on those electrons from NADH in complex 1? Yeah, coenzyme Q. Yeah, so what's happening to coenzyme Q? Is it being oxidized or reduced? Okay. Yeah, it's getting reduced to what? Yeah, exactly. So coenzyme Q is taking on those 2 electrons and also the 2 protons and becoming QH2. And then what is happening in terms of protons while all of this electron movement is happening? Yeah, exactly. And then we have 4 protons are pumped into the intermembrane space. All right, that's complex 1. How about complex 2? Where do we start with electron carriers? Yeah, FADH2. And what's happening to FADH2? Yeah, it's getting oxidized to FAD. And where are the electrons going that it's donating? Yes, same thing, exactly. So those 2 electrons are getting passed off to a coenzyme Q, and they are reducing it to CoQ and what is happening in terms of protons? Yeah, nothing. There are no protons moving. And then if we think about during our other processes that we learned about in catabolism, which complex do you think is more busy? Do you think there's more NADH passing through or more FADH2 passing through from what we learned so far? Yeah, NADH. So in general, complex 1 has a lot more going on than complex 2 because you have NADH2 coming from all these different processes we learned about. There's a lot more NADH molecules that have to go through the electron transport chain compared to FADH2. All right, so how about complex 3? Which electron carrier is entering it? Yeah. What? Both what? So they entered into complex 1 and complex 2. They passed off their electrons to something else. Do you want to go ahead? Yeah, exactly. So coenzyme Q took the electrons from them already. Coenzyme Q is carrying them now over to complex 3. So now we have our reduced coenzyme Q that took those electrons. So they're sort of coming over here to complex 3. And so what's happening with coenzyme Q now? Yeah. Go ahead. Yeah, it's getting oxidized to its oxidized form. And what's happening to the electrons? Where do they go? Yeah. Yeah. How many cytochrome Cs? Exactly. Yeah. So we had the 2 electrons from coenzyme Q. So we need 2 cytochrome Cs to take them up. And what is the oxidation state or charge state of cytochrome C going from N2? Yeah, exactly. It's getting reduced, right? It's accepting electrons. Yeah, so it goes from a 3+ state, and it's getting reduced to a 2+ state. Yeah, because of iron. All right, and then cytochrome C is now the reduced form. It's going to travel over to complex 4. And what is going to happen to those cytochrome Cs? What are they going to do with their electrons? Yeah, exactly. They give them to oxygen. So cytochrome C is going to get oxidized back as it donates away the electron. And those 2 electrons, so I'm going to write a half of an oxygen molecule, because right now we only have 2 electrons. It's going to accept them and turn into 1 water molecule. So oxygen is the final acceptor of all of those electrons that got passed along the chain there. And so yeah, and we just talked about in great detail where all these electrons are going. So something else that's also important that's happening here is our electron carriers that we made, that we need for our catabolic metabolism. They've all been oxidized back to their oxidized form. So now NAD plus and FAD are able to participate again in catabolism. So they can go through glycolysis. They can help out with glycolysis again in the citric acid cycle again, and the PDC. And the ETC is happening because we have oxygen, right? And we're regenerating these oxidized electron carriers. How can we reoxidize those electron carriers when there's not oxygen? What process do we do? Yeah, fermentation. Yeah. So when we have oxygen, we can do this. When we don't have oxygen, we can go through fermentation to help regenerate those oxidized electron carriers. Any questions about this stuff?
I don't know if you would know this, but how many times is NAD are reduced to NADH and then NADH oxidized to NAD? Like, how many cycles can it do? Does it last forever in theory?
I mean, probably eventually there are harsher environmental conditions in the cell, like reactive oxygen species can probably eventually damage the molecules, but I mean, it's just donating and accepting electrons and protons, and the rest of the molecule is fairly stable. I mean, but at the same time, it's you do like replenish it because it's derived from nutrients you taken from your diet, from I believe vitamin B1, I think. Now I'm forgetting, but one of the B vitamins. So like you do require them your whole life. So like they probably like when new cells are growing or when cells die off, you need to make more. But they're pretty recyclable. Yeah. Any other questions? Okay. So we'll go back to here where we sort of ended up. So we kind of a little bit talked about this, but why does the electron transport chain only take place under aerobic conditions? Yeah. Exactly right. Yeah. So, oops, not Q. Oxygen is the final electron acceptor. It gets reduced in complex 4, like we just talked about. Without oxygen, all of the previous pathways that we were just talking about will get backed up. The electron transport chain will get backed up, and the citric acid cycle will get backed up. Because essentially you are accumulating all these products, all of these reduced electron carriers, and they have no way to give away their electrons. So they slow down, and eventually they would probably stop, which is why we have the option of fermentation in these circumstances. Because that way, at least you can make more of your oxidized electron carriers and use them for glycolysis, which helps you make a small amount of ATP. But we're going to talk about now in the next part how the electron transport chain is a really efficient way of making a whole lot more ATP than you would get just from glycolysis. So before we do that, I just found this cute little cartoon of the electron transport chain and oxidative phosphorylation. So this is sort of, I don't know if Biochem was a comic book, this might be what you see. But just looking at this diagram, what do you see that's missing here? Or what's a little bit inaccurate? Or is it very good? Is it very accurate? Yeah. Yeah, so FADH2 isn't written there. So it does sort of, there's a nod to it because it has succinate here. And succinate turning to fumarate is what that enzyme succinate dehydrogenate catalyzes. So rather than showing the electron carrier, it's showing the metabolite, which is an interesting choice. So yeah, that was a good catch. What very important thing do we need that is not shown here? Yeah. Yeah, oxygen. There's no oxygen anywhere. Anything else anybody wants to point out? Yeah, I think you're about right. There's nothing really major missing. I mean, it doesn't tell you how many protons are generated, it doesn't tell you which side is the matrix and which side is the intermembrane space, but overall it's sort of gives you a very broad idea of what's going on. I also really liked the way that they made ATP synthase into a little hydroelectric dam at the end. So what is being represented by this water that's passing through this hydroelectric little generator? Yeah, so the water is the protons, exactly. So it's all the protons that are passing through. And as they pass through the little ATP synthase turbine, it's making electricity, which is ATP, right? All right, I guess one more question. So which side of the dam is the intermembrane space and which side is the matrix in part C? Where are the protons getting pumped? Yeah.
Reservoir side is the intermembrane space?
Yeah, exactly. So this is the intermembrane space. This is the matrix. Awesome. So if any of you go on to be scientific illustrators, you can draw cute little biochem pictures. All right. So now that we have gone over the...
Oh, they still coenzyme wrong.
They sure did, yeah.
Is the turbine supposed to be in the dilator?
Yes, actually. Yeah, we're going to talk about that next. Yeah, they actually did a pretty... Oh, that's a good point, actually. Yeah, so this part should be in the membrane. I didn't even catch that. Yeah. So the part that spins is actually inside of the membrane. Well done. All right.
Maybe the first time through just didn't go to enough biochem lectures.
Exactly, yeah. They watched like the first half of the ETC lecture and they're like, I'm set. Yeah. All right. So let's talk about oxidative phosphorylation. So, and we'll get into ATP synthase. So, first question, which is a bad question, is what makes ATP synthase a P-type ATPase? And so the first question to answer that question is, what's wrong with this question? Yeah. It's not a P-type. Yeah. Do you know which type it is? It's an F-type. Yeah. So ATP synthase is an F-type ATPase. Does anybody remember what a P-type ATPase actually does? It's from chapter 10. It's a long time ago. Yeah. Go for it. Yeah, so it moves positively charged ions, and so does ATP synthase, right? But what's the difference? Yeah. Yeah, exactly. So the P-type is going to use ATP hydrolysis to move the charged ions, the positively charged ions. The F-type ATPase, which is what ATP synthase is, takes advantage of a positive proton ion gradient to make ATP. So they kind of have the opposite function. One is hydrolyzing ATP, one is synthesizing ATP. Yeah, so I guess we actually, in that way, we sort of answered what makes it an F-type, right? So it uses a proton gradient to synthesize ATP. And does anybody know what the F stands for in F-type? So in P-type, it's for positive ions, P for positive. This is a weird one. Yeah, so for F-type, it actually refers to the names of the subunits. So there's two subunits in ATP synthase, F0 and F1. So it's an F-type, and it has F subunits, which is kind of meaningless in itself, but there we go. Okay. So which direction are the protons moving at this point when during ATP synthesis? Yeah. Yeah, exactly. So they're moving from the intermembrane space to back to the matrix where they came from during this process. So they're going down their gradient that just created, and that helps to drive this little motor that makes ATP.
I have a question on mitochondria, like anatomy. So
Okay.
would the outside membrane just be called the mitochondrial membrane?
So usually they're called the outer mitochondrial membrane and the inner mitochondrial membrane.
Okay.
So the OMM and the IMM.
Got it.
Yeah. And then there's the matrix is the very inside, the intermembrane space is between the two, and then the outside is the cytoplasm.
Got it.
Yeah. Any other questions so far? All right. So, I just wrote over my own question. So, yeah, so we just talked about what gradient is harnessed, and I wrote on top of it. I wrote the answer to the question on top of the question. It's my style. All right, so the gradient is the proton gradient. I won't rewrite it, but you all know the answer now. And it's how is the gradient generated? Where did the proton gradient come from? Yeah. Yeah. Yeah, the previous step, which was the electron transport chain. So the electron transport chain pumped all those protons from the matrix to the intermembrane space. Now they're going to all move back into the matrix, back down the gradient, and drive the ATP synthase. And then we have one last question, which we also answered already. So we know where they move now. So during ATP synthesis, they go from the intermembrane space back to the matrix, which is written over there. All right. So, yeah, so this is ATP synthase. It has a lot of different pieces. So, first of all, let's separate out the F0 and the F1 subunits. So the F1 is drawn over here, F0 is drawn over there. What is the difference between them? What does each one of them do?
I guess we'll have F0,
Mm-hmm.
transmembrane.
Mm-hmm.
That's which proton pump is.
Yeah, exactly. So this is the transmembrane portion of the ATP synthase. It is located within the lipid bilayer. It also is where the protons pass through. Do you want to add? Yeah, that part rotates. So gamma and epsilon, this rotates. And what do they rotate along with? So gamma and epsilon are this kind of rod shape over here. What are they attached to that is moving? There's only a couple options. Yeah. Yeah, so which part spins is the question. So if the protons are entering, we said, from the intermembrane space, they're passing through which part? Yeah. Yeah, so which piece of the F0?
or one, N1, P2, C12.
Now you're just naming all the pieces of F-Zero.
Yeah. Oh, yeah. Okay.
So let's start with the bottom, basically. So the protons are entering from the intermembrane space. So are they entering through A? Are they entering directly into C? Are they entering this little B piece? Where do they go in? Yeah. Yeah, they enter through A. So the protons enter through A, but before they can exit, where do they pass there? C, exactly. So the little yellow, they look yellow for you too. Yeah, the little yellow circles in the C ring are the protons, or depictions of the protons. So they enter in through A, they enter into the subunits of the C ring, and because protons are charged, they change the overall charge of that portion of the C ring, and that helps it want to move deeper into the membrane, into the nonpolar lipid part of the membrane, because it makes it more hydrophobic. So as the protons enter through A and pass through C, they're causing this rotational movement as they push each segment of the C ring further into the lipid bilayer. So the C component is a ring. You can call it the C ring, and it rotates as the protons enter. And so essentially what's happening is this electrochemical gradient that we have from the protons is being turned into mechanical energy because it's physically pushing the C ring through the membrane. And then eventually the proton on that C subunit will, as protons keep entering, once the C ring makes one full turn, the proton ends up back at the A subunit and it can exit on the matrix side. So it goes around in a circle and then it comes back and then it leaves. And so that covers the F0 subunit. So what about the F1 subunit? What is its main job? Yeah, so this part is actually making the ATP. And which side of the membrane is it located on? Yeah, the matrix side. So we just mentioned that the gamma and the epsilon subunits are attached to the C ring. So as the C ring rotates, they also rotate. And then what's going on with these other subunits? Now that we have this rotational movement, how are we turning that into ATP?
It's like ABP5 enters from the alpha and beta part.
Yeah, so is it the alpha part or the beta part that is actually doing this catalysis? Yeah, the beta part. So beta is turning ADP and inorganic phosphate into ATP. And what about the other ones? What do alpha, delta, and beta do? And B, sorry, not beta. Do they have a function in catalysis? Seeing some no's, that is true. So they are not actively catalyzing this reaction, but they're really important because they are structural components. Yeah, they hold the whole enzyme together. And they remain stationary. I also want to emphasize that even though we have these subunits called alpha and beta, that does not refer to their secondary structure. So kind of like with hemoglobin had alpha and beta subunits, even though their secondary structure is mostly alpha helical. Same thing here. Alpha and beta refer to their names. It's not their structure. I mean, you can see here there are a lot of alpha helices and some beta sheets, but alpha and beta are mixtures of both. That's their name. It's not what they're made of. Any questions about that? Yeah. Yeah, they're basically, let me just go back to that picture from the top down, but this picture doesn't show them, right? Yeah, so here's the top down picture. So they're basically spaced with one another. So the purple ones here are alpha, the yellow ones here are beta, and they're, yeah, I don't know how to say it other than like the ATP is formed in these three. So these three are going to have a conformational change that happens as you get that rotational movement. The alphas, I think they also do change shape, but they don't actually catalyze the reaction. And they help keep everything together and hold everything in place, but they're not directly catalyzing the reaction. Oops. Okay. So, yeah, so I think that covers the structure and the function. So we have this kind of bluish part at the bottom that's rotating, that's helping to cause a conformational change in the greenish part that's in the matrix. And that is ultimately helping to catalyze the formation of ATP. So for every one complete turn of our C ring, we get one sort of round of catalysis. We have 3 beta subunits. So for every complete turn, each of those beta subunits makes 1 ATP molecule. So for every complete turn of the C ring, we get 3 ATPs that we make. So the question you might have is, how many C subunits do you have? How many protons do you need to make 1 complete turn of that C ring if every subunit accepts 1 proton? So it wasn't a lecture. So how many C subunits do we have? 10. Yeah. And so in this diagram it says 10 to 15 because different organisms have different numbers. We have about 10.
So
Yeah.
if you have more C subunits,
Yeah.
then you require more protons to create one ATP.
Exactly. Exactly. Yeah. The more subunits you have, the more protons are needed for 1 full turn to make 3 ATPs. So yeah, in a sense, having more is less efficient. Yeah. Any other questions so far? Yeah, exactly. Yeah. Every one turn, it's 3 beta subunits make 1 ATP. So 3 total. So that brings us to more math. So yeah, so let's start filling this in. So at this point, now we know enough about how everything works to start calculating from the very beginning, starting with eating sugar and getting glucose out of it, to how many ATPs you actually make from it. So for NADH, so we just chatted about in the electron transport chain, how many protons do we get from one NADH that get pushed into the intermembrane space? Yeah. Yeah. 10. So every NADH gives you 10 protons. And then we have, the way this is written is a little bit confusing. I'm going to rewrite this in a different way. So we have 10 protons. So you can also write this like that, so that you can eventually cancel out the units. So we're essentially just doing dimensional analysis, where we're doing some math to cancel out units to eventually figure out going from protons to the number of ATPs we can make. So NADH gives us 10 protons. And so on our ATP synthase, we just talked about one turn, or sorry, that's very, I think I just wrote this wrong, sorry. That's what I get for being in a hurry. Like, this doesn't make any sense. And I just confused myself. Yeah, I definitely was right. Losing my mind. Okay. Yeah, so in our C ring, one turn requires how many protons? 10. Yeah. And for every turn, for every complete turn of our C ring, how many ATPs? Yeah, 3 ATPs. There we go. All right. So if you do all that math, what is the final number of ATP that you get? Yeah, 3. This way this way everything cancels. Okay, great. So that's our theoretical number, right? We have 10 protons per turn. We have 3 ATPs per turn. We make, theoretically, 3 ATPs. That's the sort of ideal maximum number you can get. And that's the number you can get by calculation. In real life, we don't get quite as many as you theoretically get. So does anybody know the actual number of ATP you get for one NADH? Yeah. It's more than that. Yeah, it's about 2.5. So that's not a number you can calculate. That's a number you have to remember, because the theoretical one you can calculate. The actual, it's a little bit less. So why is the actual number a little bit lower than the theoretical number? What's happening to all the protons? Yeah, exactly. So the membrane itself is a little bit leaky, so some of the protons can go back to the matrix without passing through ATP synthase. Any other reasons? Yeah. I'm sorry? Yeah, I mean, that's sort of the idea behind like a leaky membrane. It's like non-optimal conditions. So in real life, some of the protons go somewhere else. Yeah. Yeah, that's also true. So yeah, not all of the energy of the electron transport chain is put into pushing protons. And also, once you made a proton gradient, other enzymes can take advantage of the gradient to drive their own reactions that are not ATP synthase. So not all of the protons that you pushed into the intermembrane space are going to pass through ATP synthase, either because other enzymes are using them or because they leak through the membrane. Okay, so we did, yeah.
But you said that you have to remember the actual.
Yeah, so this number, the 2.5, is not a number you can calculate. Like, you can figure out the theoretical number by doing this math we just did. The actual number is a little bit less because it's not a 100% efficient use of all the protons. Yeah.
How does the NADH with the 10 protons start?
So that's because of the electron transport chain. So it goes through complex 1, 3, and 4. Complex 1 gives you 4, complex 3 gives you 4, complex 4 gives you 2. So 4, 4, and 2 makes 10. Yeah. So how about for FADH2? How many protons do we get from there? Yeah, 6. Yeah, so FADH2 is skipping complex 1. It only gets the protons from complexes 3 and 4 because complex 2 doesn't give you any. So we only get 6 protons from the electron transport chain. And then for our C ring, how many protons give one turn? Sorry. Yeah. It's still 10. Yeah. And how many ATP do we get for every one turn of our ATP synthase? Still 3. So now if you do this math, how many ATPs does that give you? Yeah, 1.8. So our theoretical number is 1.8. And so is our actual number higher or lower than our theoretical? It's lower. Yeah. Does anybody have that number? Yeah, it's about 1.5. So if you were able to track each proton that was made by each of these electron carriers, that's what you would get. In reality, once you pump all the protons into the intermembrane space, they're just protons. You can't really tell, oh, this NADH gave me these 10 protons that are all going into this ATP synthase enzyme. It's protons are protons, but that's where they all come from. And just to be clear, something like this will be on your exam, so be able to do this math and know these numbers. Any questions about that? Yeah. Why the what? So yeah, so basically, if you could use every single proton that was pushed into the intermembrane space, and if you use every single one of them and put them back through ATP synthase, then you would get that theoretical number. But you lose some of those protons. So some of them just leak back through the membrane without passing through ATP synthase. So they just go back into the matrix by themselves, some of them. And some of them can also be used as a gradient source for other enzymes to catalyze other reactions. So you have a proton gradient, it drives ATP synthase, but it might also drive other reactions.
Wait, so it's the same reason for NADH?
Yeah, because protons are protons, right? Yeah. Any other questions? All right. So now it's time for more math. So we love a table. So we have our three different catabolic processes: glycolysis, the PDC, and the CAC. And then you can start putting together for every glucose molecule, so this is per glucose, how many ATP or ATP equivalents, such as GTP, do you make directly? How many NADHs do you make? How many FADH2s do you make? And then for every NADH, how many ATPs do you make? And for every FADH2, how many ATPs do you make? And then we can add them all together. So before you begin, let's just review. For every NADH, how many ATPs can you get at? Actual, yeah, so 2.5. And for FADH2, 1.5. So now that you have that information, go ahead and take a few minutes and start filling in this table. All right, let's do some math. So let's start with glycolysis. Does glycolysis give you any direct ATP? Yeah, you get 2 ATP for every glucose. And how about NADHs from glycolysis? Yeah, 2 NADHs. So let's skip over to, we'll do the math after, so let's skip over to FADH2s. How many FADH2s do we make from glycolysis? Yeah, 0. Okay, so let's fill in the PDC now. So how many PDCs, how many ATPs do we get directly from the PDC? None, yeah. How many NADHs? Per glucose? 2, yeah. How many FADH2s? 0. All right, how about from the citric acid cycle? How many ATP equivalents? 2. Yeah, we make those 2 GTPs. How about NADHs? 6. And how about FADH2s? 2. Great. So does anybody have questions so far about where these numbers came from? Awesome. All right, so then now we can multiply everything times 2. Or not times 2, times 2.5 in the case of NADH. So 2 times 2.5 gives you 5. 2 times 2.5 still gives you 5. 6 times 2.5 gives you 15. For FADH2, 0, that's very easy. And then 2 times 1.5 is 3. And then we just have to add everything up. Just going to make these red so they're more obvious that we also have to add them together. So then we can just add together all of our ATPs. Those don't look this different to you at all. These are completely different colors on my screen. I apologize. All right, so we'll add together all of the ATP equivalents. So if we go across for glycolysis, 2 and 5 and 0 gives you 7. For the PDC, 0 and 5 and 0 gives you 5. For the citric acid cycle, 2 and 15 and 3 gives you 20. And then if you add together going down this way, get 25, get 3, oh boy, so much math, get 10, 2, all right. So what's our total number of ATPs that we got? 32, yeah. And so, yeah, so we calculated 32. So for sometimes in other sources, you'll see slightly different numbers. Like in that cartoon we saw a few minutes ago, it had 34. Sometimes you'll see sources that just say more than 30. And this is partially because the actual versus theoretical numbers, depending what number you use as your actual number, it can change the total a little bit. So if you do it this way, you get 32. That's what I care about. But if you have other textbooks or look at other sources, you might see slightly different numbers. And I just want to also emphasize you should be able to do this on the exam with these numbers. Does anybody have any questions about, yeah?
So just to make sure, whenever you say ATP equivalent, you gotta like refer to like GDP or ATP.
Yeah, so any nucleoside triphosphate that can be used as an energy currency. So yeah, so generally ATP, GTP, UTP for the purposes of what we've done this semester. It's for glycogen synthesis, yeah. Yes. Any?
So like directly,
program glycolysis where we do directly produce for ATP, but then the next product is just
Right.
So I personally use for direct.
Okay.
And so I was wondering if like direct means like forget about the enzyme. What is the actual form of ATP that we're producing?
I see what you're saying. Yeah. So yeah, so like if I would say for that you would say gross versus net. So here direct for me at least, or I would say that means like substrate level phosphorylation. So like you're which I mean it's true you're making those for, but when you're saying like per glucose molecule, it's sort of what are you getting out of the whole process of glycolysis, if that kind of makes sense. If I asked you like in stage 2 how many do you get, but if you kind of total together glycolysis as a whole, you got to subtract out the 2 that you used, yeah, because that's how many you get at the end. Any other questions? All right. Well, that's all the questions I have for you today. So I just want to give one last shout out to Dr. Middleton, who trusted me to teach her class all semester. If you ever want to get in touch with me about anything when I'm no longer at Purchase, this is my personal email if you, for some reason, need to talk to me after this. Here's a terrible joke about polar bears for your enjoyment. And then just as a reminder, I have office hours today. Next week, most of my office hours are virtual. It's all posted on Brightspace if you want to talk to me before the exam. If you haven't done the last quiz yet, that's due tonight. Your semester-long projects are due tomorrow. And your exam 3 is right here next week at noon. So usual class time is 12:30. It's earlier. It's at noon. So if nobody has any more questions, we can end it there.
Thank you very much.
No problem.